Good afternoon, everybody, and welcome to this webinar from the British Society for Immunology. Fantastic to have you here. We've got hundreds of you signed up, joining us this afternoon, which is really, really wonderful. Some of you may not have engaged, in fact, with the British Society for Immunology before. So a quick, in a nutshell, who we are. First of all, I'm Doug Brown, I'm Chief Exec of the British Society for Immunology. And essentially, we are the organisation that represents everything to do with immunology in the UK, but also with some global reach as well. And a big part of our work that we've been focusing on for a number of years is around advocacy, and particularly advocacy in the vaccine space, where we have worked on childhood vaccinations, working in partnership with other organisations and professionals in the field to try and increase the uptake of childhood vaccinations across the UK. Of course, since the pandemic has hit in the last year, we've been really active on COVID immunology as well. And of course, a big part of that is around COVID vaccinations, which of course is the theme of today's session. And I'm really delighted that we've got two amazing speakers with us here today, have given up their time to speak to you out there across the UK about vaccination, about immunology, about COVID vaccines in particular. And so they will give short presentations, just 5 minutes or so each. And then the rest of the session is open to question and answer. So please do submit your questions via the chat function that you can see on the webinar here. Those questions will come in centrally, and I will ask as many of those questions of our speakers as possible. So get your questions in, the sooner the better, and more likely you will be able to have your question asked and answered. Oh, I just had a flag saying that I may be experiencing network connection difficulties, so apologies if that has been a little bit jumpy. Hopefully that will go away. And so one thing we did want to do in the setup here was to give a bit of a plug to a very new resource that has been released and launched by the BSI. That is here, our guide to vaccinations for COVID-19. And in fact, we just released it this week, working with many people in our membership, including those on the call webinar today. And it's your go-to guide for lots of information around the COVID vaccination. So please do, if you haven't seen it already, do download it from our website. It's all over our social media, but it's on our website as well. It'd be great for you to have a look. Don't worry about jotting that website down now. I will show this again at the end, and it's on the front page of the website, the British Society for Immunology. So that's the BSI plug out of the way. I'd now like to bring us to our speakers. And as I say, we will have a short presentation from each of our speakers. Our first speaker will be Dr. Emma Chambers, who is from the Blizzard Institute at Queen Mary University of London. Emma is actually a trustee of the British Society for Immunology as well. She does a lot of work for us and is one of our vaccine ambassadors, and is fantastic at engaging with members of the public and healthcare professionals on this vaccine topic. So great to have you, Emma. Very much looking forward to your talk. And our second speaker, panelist today, is Dr. Brian Ferguson, who's from the University of Cambridge, who actually does his research on vaccines as well, and again has helped us immensely with lots of resources around public engagement and educating healthcare professionals, and is also one of our vaccine ambassadors as well. So I can assure you, you have two of the very best people in their field and able to explain complex scientific topics in an understandable way as well. So no pressure to our speakers after setting them up in that way. So I think we will now move to our speakers. I am also going to be the equivalent of Chris Whitty's assistant this afternoon, because I'm in charge of the slides. So speakers, when you'd like me to move to the next slide, please do request next slide, please. And I'll take you through the presentation. But I will leave it there. And Emma, I will hand over to you to take us through the first part of the presentation. Emma, yours.
Great. Thank you, Doug. Next slide, please. Next slide, Doug. Good. So to begin with, I'm just going to talk about what is immunity and how does the immune system work. Oh, sorry, can I have the previous slide, please, Doug? Thank you. And really describe what it is it does and which cells contribute. So first of all, what is immunity? What is an immunologist? Well, so the immunity is basically our natural biological defense, which prevents infections from bacteria, fungi, or viruses, fights disease. We know that we now use the immune system to fight off cancer, and also prevents unwanted biological invasions. So we have the barriers of our body which prevent infections, such as the skin or the gut or the lungs. Next slide, please. But the main cells which are used to fight off any invading pathogens are our white blood cells, which are known as the immune system. And here I've shown you a few examples of white blood cells that are commonly found in the body. If you click next slide, please, twice, I think it will bring up everything. Perfect. So we have the innate immune system, which is made up of white blood cells called things like monocytes, eosinophils, basophils, and neutrophils. And these are our first line defense. They're the ones that when they see something which isn't them, they immediately respond to it. And then they send out signals which instructs the other members of the immune system to come and help fight it off. And they're known as the innate immune system. The other cells which are important in our immune system are the ones which form memory, such as the T cells and B cells. And these are known as the adaptive immune system. What they do is they go to the sites of where there is an invasion of bacteria or viruses, and they grow and get bigger and eventually form memory. And these are the cells that vaccines target to give the body a head start when fighting off infections. Next slide, please. So one of the key questions is how does the immune system work? And here I'm showing you an example of what happens with B cells, but a very similar immune response happens with T cells. So we have our first infection, which you can see on the left hand of the slide, where you encounter your pathogens, so your bacteria or viruses. And then the naive, the young B cells go to the site of where this infection is. They get activated by the signals that are present there, and then they grow up, get bigger. And then at about 7 days after exposure to this virus or bacteria, they start producing lots of lovely antibodies which are used to fight off the invading bacteria. And as a result of this peak of the first primary immune response, you then get a reduction in the virus or bacteria that's entered you, and the B cells have done their job. What's the other lovely thing about the B and T cells in our body is that they then go on to form memory. So they reduce a bit in number, and they go and live in parts of our body where they form a long-lived memory and wait for exposure to the bacteria or virus again. So then when you get a repeated infection, as shown in the second part, these memory cells are there, present, and ready to respond quickly and more efficiently, a much quicker immune response than the first line of infection, and therefore can fight off the invading pathogen, bacteria or virus, much quicker. And this is really what we use when we generate a vaccine. So next slide, please. Thanks. So basically, the vaccine is using this clever memory response of our immune system to generate a memory response in our body so that we're ready to fight off the pathogen again when we next encounter it. So what happens with vaccines is you inject a small part of the virus or an inactivated part of the virus into the arm, which activates your immune response and initiates a memory response from your adaptive immune cells, so your T and B cells. What happens with the B cells, as I discussed earlier, is they make very specific antibodies, which are then circulated in your body and also generate memory B cells, which sit ready to fight off the infection next time it encounters it. The T cells are very similar to B cells, apart from they are cells which have the capability to kill infected cells. And they expand in response to this vaccine, and they then go and form a long-lived memory, and therefore they're ready to fight off any invading virus or bacteria again. These cells basically remember the virus and they live in your body, and this is known as immune memory. And this is the key component of how vaccines work. Therefore, if you encounter the real virus again in the future, your body's ready. It's ready to fight it off with its memory cells, and initiates a really quick, rapid immune response, which can fight it off much quicker and prevent a serious infection. What I'll do now is I'll hand over to Brian, who will discuss the vaccine specific for COVID-19.
Thanks a lot, Emma. That was great introduction. Hi, everyone. I'm Brian. Thanks, everyone, for joining the webinar. I'm just going to give a brief introduction to the specifics of COVID-19 vaccines, the ones that we're using in the clinic in the mass vaccination campaign that's going on in the UK at the moment. But it's worth just remembering, as you can see on this slide, that 12 months ago, in January 2020, was the first time we've had the genetic sequence of the SARS-CoV-2, this new virus that causes COVID-19 disease. And in the 11 months between January and December 2020, we went to a stage not knowing what that virus was, what the genes were like, what the genetic sequence was, to a stage where we were starting a mass vaccination campaign in the UK with a vaccine that has a greater than 90% efficacy against the disease. And that's an extraordinary story of human endeavor and collaboration and skill and efforts that went into getting to that stage that we're at now, where we, and especially those of you involved in the mass vaccination campaign, can use these vaccines to help us to really defeat this virus. Next slide, please, Doug. So in the UK, we did a very good job of procuring several different vaccines. We're lucky enough that two of those vaccines, the Oxford AstraZeneca vaccine and the Pfizer BioNTech vaccine, went through the phase 3 trials relatively rapidly and were approved by the MHRA regulatory body, so that they could get rolled out into the clinic at the end of last year. And those are the ones that I'm going to talk about a little bit more detail today. The Oxford AstraZeneca vaccine is an adenovirus-based vector vaccine, and the Pfizer BioNTech vaccine uses the mRNA technology, which is relatively new, but has been in development for a couple of decades as a technology, as a concept. We're also in a position now where in the UK where the Moderna vaccine, which is another mRNA vaccine, has been approved for use, although hasn't yet been manufactured and delivered to the UK, so won't be in the clinic until we receive those doses sometime in the spring. And just recently, the Novavax and Janssen vaccines have also passed the phase 3 trials and look great, especially the Novavax vaccine, which looks fantastic. That will be hopefully going through regulatory approval soon, and will be manufactured in Teesside. So we'll actually join, fingers crossed, we'll join the other vaccines in the clinic for the vaccination campaign relatively quickly. Next slide, please, Doug. So just to say a little bit about the Pfizer and the Moderna vaccines. So these two are based on mRNA technology. And what that means is that they have a genetic code which carries the instructions to deliver to our cells to allow our cells to make the SARS-CoV-2 spike protein. And it's that spike protein which is the target of antibodies that are able to neutralize the virus and stop it from infecting our cells in the future. So this is a great way of training our immune system to see something as a danger, and then to remember its shape and to make antibodies against it, which will stop future disease. And so both of these vaccines use the mRNA technology, and that's a piece of genetic material, which is then coated in a lipid coat to make these tiny particles, which are called nanoparticles, which just means they're small droplets of lipid which have this genetic material inside. Following that production of those lipid particles, they're injected into our muscle in 2 doses, and inside the muscle tissue, and the cells inside the muscle tissue then start to synthesize using that genetic code, the SARS-CoV-2 spike protein. The spike protein is then presented to our immune system. Our immune system sees it, remembers the shape, and makes the antibodies against that protein, which then can protect us against future infection. Next slide, please, Doug. So how do we know these things work? Well, so there's two main ways that we know they work. In the phase 1 and the phase 2 trials of the mRNA vaccines, and this, what I'm showing here is just some data from the phase 2 trial of the Pfizer vaccine. The people carrying out this trial could take blood from patients on the trial who'd been inoculated with the vaccine, measure the amount of antibodies that were produced in the blood of those patients, and take that blood and see exactly how good those antibodies were at stopping the SARS-CoV-2 virus from entering our cells. And that's what this data is on the screen here. And you can see in the red boxes that people who've been injected with that virus were making very high levels of neutralizing antibodies, very high levels of concentrations of antibodies in their blood, particularly at 28 days after the first dose. And that's really what that means is after they've had 2 doses, they have very high levels of antibodies, which are a very good sign that this vaccine was going to help to protect against disease. So for the next slide, please, Doug. And that could be seen then into the phase 3 trials by looking at counting the number of people who were either injected with PBS, with saline, or were inoculated with the with the vaccine, and counting in those people the number of people who then went on to develop COVID-19 disease. And you can see in this graph here that the people who've been injected with PBS, they, on the red line, a lot of those people went on to develop COVID-19 disease. And the people who've been vaccinated, in the blue line, very, very few of them went on to develop disease. And this is an extraordinary result, actually, showing that this vaccine almost completely protected people against COVID-19, and that's the reason that it was approved by the MHRA and is now available in the clinic. And it's worth also saying that those people who had been vaccinated and then went on to develop COVID-19, their disease was very mild. None of those people had to go to hospital. So none of those people who've been vaccinated and then caught the virus had severe disease or had to go to hospital. So not only does this vaccine protect the vast majority of people against getting ill with COVID, it almost completely protects people against having to go to hospital with COVID. And that's a very important point that I'll come back to later. So can you press the forward slide, please, Doug? And this is just a little bit further information about that, saying that most of the COVID cases occurred in the people who had been vaccinated, occurred within 14 days of the first dose. And that's really before the antibodies had been given a chance to develop in their blood. So after 14 days, 21 days, really the vast majority of people are very well protected against COVID. Next slide, please, Doug. And there was a very similar bit of data from the phase 3 trials of the Moderna vaccine, which showed almost exactly the same thing, which is that the vast majority of people didn't get COVID-19 disease after being vaccinated. And so the efficacy of these vaccines is 95%, which is absolutely extraordinary. And a great advert for this relatively new type of technology. Although it's been developed, having been developed for a long time, these are the first 2 mRNA vaccines that have made it to the clinic to help us protect against a particular infection or disease. So next slide, please, Doug. Now, the other vaccine that's being used in the UK is the AstraZeneca or the Oxford vaccine, as it's being known. And that works in a different way. So it's using an adenovirus vector, and the adenovirus itself has been modified again. So it has the genetic material that allows our cells to make the SARS-CoV-2 spike protein, and this adenovirus has also been modified so that it can't itself replicate itself in humans, in people. And that means it's very safe. It's no longer an active virus, but it can be used to deliver the gene, the instructions that make the SARS-CoV-2 protein once inside our cells. And then in exactly the same way as the mRNA vaccines, once it's been injected into our muscle tissue and gets inside our cells, those cells then make this SARS-CoV-2 spike protein. Our immune system sees that protein, makes antibodies against it, and helps to protect against infection. So next slide, please, Doug. And this data here is equivalent data to what I just showed with the mRNA vaccines. So in the red box there, you can see that people who have been given the SARS-CoV-2, sorry, the Oxford AstraZeneca vaccine, after 14 days, between 14 and 28 days after inoculation, after vaccination, they develop very high levels of antibodies in their blood, and those are the antibodies that are protecting against infection. So again, this is a really good sign that this vaccine, like the Pfizer vaccine, was going to give us protection from disease. So next slide, please. And this is again a similar graph to what I've shown for the Pfizer vaccine. The phase 3 trials of the Oxford vaccine were very, very promising. So the vast majority of people who got the vaccine were not then going on to develop disease. And in fact, in the same way as in the mRNA vaccines, none of those people had to go to hospital, and none of those people had severe disease after vaccination. And that's another remarkable result. So showing that all of these vaccines are extremely good at protecting people against having to go to hospital with COVID. And that's a very powerful message, I think, to send people. The Oxford AstraZeneca vaccine has some advantage over the Pfizer vaccine. Now, the Moderna vaccine is in that it can also be stored for a longer time in the fridge at 4 degrees centigrade, which makes the distribution easier and makes it easier to look after it until the point that it's being used in the clinics. So next slide, please, Doug. So just finally, to sort of summarize all of that, these vaccines, they're absolutely amazing. They work brilliantly. They protect basically everyone against having to go to hospital with COVID, and themselves have extremely good safety profiles. So as medicines, they're extremely safe. There's very, very vanishingly rare severe adverse effects from these medicines. And so that makes the risk-benefit calculation of taking these vaccines pretty easy to make, in the sense that there's very few adverse effects. There's almost no severe adverse effects. Whereas the chances, if you compare that directly to the chances of getting really ill, getting long COVID, or even dying from COVID, it's much, much higher chance of that happening than compared to any risk of taking the vaccine. So that's the kind of message I think can really help people to understand why it's a powerful medicine to take. So I'm just going to hand over back to Emma now, and thank you very much.
Thanks, Brian. So I just didn't do one brief slide about how to speak to vaccine hesitant individuals. Thank you. You know, most people aren't really angry anti-vaxxers. They're just worried about the vaccine and about side effects, and they don't know where to get the right information from. So I think it's really important to acknowledge their concerns. There are going to be side effects to having the vaccine, such as a raised temperature or a raised bump where you're injected. And it's important to be just honest and say, yes, this does happen, but it's an example of your immune system working well and recognizing the vaccine and responding to it how you want. The other thing is, with the rise of social media, there's just an awful lot of misinformation available. And they just don't know where to get the right information, which is why we as a BSI try to put out as much easily accessible information as we can. And we provide booklets and leaflets, and our website is available for everyone to look at. And they just want to know what's best for themselves and for their families. So really, it's important just to be honest and to reassure them that the vaccines are safe, and that They are the best option available at the moment to fight this current pandemic. Okay, so I think we've finished now, and I think we'll then answer any questions that have been put in the chat box.
Brilliant. That was fantastic. Oh, Brian, we have you back, which is good. Thank you very much, both. Two fascinating and very complimentary talks as well. And we've had some really good comments coming in from people on the webinar saying that they're loving it so far. So that's good. Well done. And thank you everyone for watching, because we've had huge numbers of questions coming in. So you're making my job as chair very easy. So thank you very much for that. So let's kick off straight into the questions that have been coming in. And we've had quite a few coming in talking or asking questions about, well, if you've already had COVID, do I need the vaccine? How long after having COVID should I wait before having the vaccine? So lots of questions coming in from patients along those lines. I mean, Emma, Brian, I don't know who wants to have a go at these first, but the key ones that are coming in: if I've already had a COVID, do I need the vaccine? If so, is one or other of those two available? The best one at the moment. How long after having COVID should I wait for a vaccine? And somebody was saying that they had a patient that had a first dose, then got infected a couple of days later from the virus, and had questions around, well, when is the best time to have the second dose, as that has happened, and will that affect vaccine effectiveness? So any insights into any of that from either of you?
Do you want to start, Brian?
Yeah, I can, it's fine. So whilst you have symptoms, it's not a good idea to take the vaccine. I think waiting until waiting till your symptoms have gone away is much better. So not whilst you're sick, and so that's a standard advice for people getting any vaccine, is that they would ask to wait until they feel better before getting vaccinated. So that's a general piece of advice. In terms of people who have had COVID and then asking whether to get a vaccine or not, the answer is at the moment. And then actually there was a couple of publications came out yesterday about this, showing that people who have had COVID and then been vaccinated a month or longer later have extremely good responses to the vaccine. It's like a boost to their immunity, and that seems to be a very powerful way to generate long-term protection. So I think the advice would be yes. The advice is yes to get the vaccine, but to wait till your symptoms have subsided first.
Brilliant. Anything to that's great, thank you, Brian. Anything to add, Emma?
So my internet cut out a bit. So did Brian answer the bit about the fact that some people might get the COVID-19 after they've had the vaccine?
No, I didn't answer that.
Okay, cool.
A little bit. So yeah, you can elaborate on that.
So I mean, I think Brian's graph that he showed was a really good example in that the body takes a couple of weeks at least to generate, a really good immune response after the first vaccine. And in reality, a full vaccine course is 2 doses. So that means that it's always the chance that someone who had the vaccine and then was exposed to someone else might get the disease, within a few days afterwards. And that's why it's really important to try and still distance and socially distance and continue with the current lockdown that we're doing, because it takes a while for your immune system to get going. And that's just really unfortunate. It does happen, unfortunately.
Great, thank you both. Hopefully that answers the questions that have come in. We've had a few more questions on the kind of vaccines and how they work specifically. So one was, these healthcare workers are getting questions around the different strains, the variation viruses that are coming through. Is there any evidence to show that the vaccines will be less effective or the same for these different variants that are emerging?
There's a little bit of evidence which suggests, just in some very preliminary experiments, which suggest some of the antibodies that are made by the vaccines may not be quite as good at binding to some of the variants that are coming through at the moment. But there are two caveats to that. Firstly, those experiments haven't been done in people or animal models or anything, so these are very preliminary investigations which need to be carried out really in the context of a whole immune system. It's also worth saying it's very, very likely that the current circulating variants will still, the vaccines will still be effective against those variants, because there are lots of different ways in which these antibodies are being produced to bind to the spike protein and stop it from being able to enter cells. So our bodies make lots of different mechanisms of knocking out these spike proteins, and so just changing one little bit of the virus is very unlikely to make the vaccines not work properly at all. So it's there's some high confidence that they will still work, hopefully to the extent of they stop people getting sick, but that's not entirely known yet.
Yeah, I think it's just important to reiterate that the vaccine doesn't just make one antibody. It makes many antibodies across the whole, protein that it's exposed to. So just a few mutations just isn't going to stop the vaccine working necessarily. It might just make it a little less efficient, but we hope that it's still going to reduce severe disease and hospitalization.
Brilliant. Very reassuring. Thank you. And some people have come up with questions that they're getting again from their patients that, we're using some of this quite scary scientific terminology like messenger RNA vaccine, and that can scare quite a lot of people out there. So what the question is, what are some of the reassuring ways that people can explain why being injected with messenger RNA isn't a danger? Emma, is that something you want to take first?
Yeah, I think the first thing to show is that we've had over 9 million doses handed out, and they might not all be the Pfizer vaccine, but a large proportion are. And we've had very few serious adverse events, and those adverse events were anaphylaxis in individuals who had likely to have allergic reactions to things. So I think the first thing is to reassure everyone that there has been a lot of vaccine handed out already, and that it's been safe. And the other thing is that they've been rigorously tested by the companies, and then we've also had the approval from the Medical Health Research Association, MHRA, I've forgotten what it stands for. And there's been a lot of organizations and companies and the government have looked at these vaccines and are just confident that it's safe. And I think the evidence so far for the past month really reiterates this as the point. And yeah, there's a lot of scaring about the language used, and I think us as scientists needs to work a bit better about the way we communicate our science, certainly, and that's something that we're going to hopefully try and work on. And hopefully the booklets that we've got available on the BSI website will also reassure people.
Great, thank you, Emma. Anything to add to that, Brian?
Not specifically, no. I mean, everything that Emma said is absolutely correct. There's so much safety data now about these vaccines. None of the safety aspects that any new medicine would ever go through have been shortcutted or missed out or anything for these vaccines. It's all been done rigorously, and they wouldn't be approved without that information. It's extremely understandable that people are a bit nervous about the terminology because it's new. There's a new type of medicine, so it's intrinsically a bit frightening for people. That's absolutely understandable. But hopefully, the fact that as more and more people go through the trials or have been given these vaccines, and we see that there are vanishingly rare side effects or vanishingly rare, especially severe side effects, that people want to see that they're really safe.
No, definitely. And on that point about not taking any shortcuts in the development and testing them thoroughly, there have been a couple of questions that have come in that people are getting from their patients, but also from staff and co-workers in hospitals and in surgeries about, well, how has this vaccine development process been sped up so much. Usually it takes 8 to 10 years to develop a vaccine, and we've got a whole host in less than a year. So there is a job that needs to be done in winning hearts and minds around that. But what reassuring words can you give, or explanations as to, well, how have we been able to do this so quickly? Brian, did you want to start, and then Emma? It'd be great to hear from you.
Sure, yeah, that's no problem. So there's various aspects that have allowed that process to progress relatively quickly, or compared to previous vaccine development stages. First, I think it's worth saying firstly, and I sort of tried to get this point across in my talk, is that there's been a building of many decades actually of basic scientific research that's gone into making these vaccines. So it's not starting from scratch, it's using information and knowledge and scientific research that's been happening for many years, building on top of that to now apply that to this problem of how we're going to tackle SARS. So we weren't starting from a baseline of 0, actually. It was starting from quite a high level of knowledge about how coronaviruses work in general and how to make vaccines. So that's the first thing. The second thing is that there was a lot of upfront funding for this vaccination development, which allowed things to progress very quickly. Scientists globally didn't have to suddenly write lots of grant applications and spend many years waiting for the funding to come through so they could start doing everything. It was funded very quickly and to a very high level to allow everything to kick off fast. Another thing is that that funding allowed the manufacturing process to start whilst the trials were going on. Whereas usually what would happen is that all the trials data would be generated, and then the manufacturing would start after that. But the manufacturing process was begun during the trials because there was money to do it. It was a risk, but the risk was taken away by the funding from private sources or government to allow that to happen very quickly. Another thing is that there were so many people signing up to the trials all over the world, working together to try to step forward and say, yes, you can test me with these new vaccines. The trials started really fast because they had enough people to start the work, which was incredible. And then the final thing is that there weren't many years of testing in preclinical models, so working out if it was going to work in the lab first. They went very quickly to the human trials because there was such an urgent need for the vaccine in people. So all of those things together meant that it could go very fast, and I'm sure there are more things to add to that, but those are just some of the reasons.
Brilliant. So yeah, definitely lots of barriers taken away and lots of funding to facilitate the process. That's fantastic, Brian. Anything to add, Emma?
I mean, not really. Everything Brian said is correct. We knew a lot about coronaviruses before, and we knew a lot about vaccine design before. Money wasn't an issue, which used to be the main stopping step of any vaccine development. We had so many people volunteer for the trials. I know a lot of people personally who took part in the Oxford trial, and also our country had a lot of disease, which meant that we were able to show quite quickly when the vaccine was able to work. Yeah.
Brilliant. No, an absolute global Herculean effort, which is continuing as well. So fantastic we've had these vaccines so quickly. So changing tack slightly, we've had quite a few questions coming in about dosing and gap between first dose and second dose. And of course, the UK moved to a slightly different dosing schedule, which was different to that tested particularly with the Pfizer vaccine, different to tested in the clinical trial. So the questions are, well, what level of effectiveness does it look like people will be getting after one dose? And will there be any impact on immunity if there is that maximum 12-week between first and second dose? Who would like to answer that question?
Do you want to kick off, Emma? I can
yeah, I'll give it a go. So, you know, in an ideal world where we had no coronavirus cases here, we would have, of course, wanted to keep to the vaccine schedule that was designed as per the clinical trial, where we give 2 doses within, I can't remember how many weeks it was now. But due to the current situation in the UK, where we had a colossal number of coronavirus cases, COVID-19 disease and hospitals were full, the best way to have the biggest impact of vaccination was to slightly increase the time between the first and second dose. We as a BSI think it's important to, first of all, make it clear to individuals that 2 doses are necessary to have a full and broad immune response. And we need to make sure it's clear to everyone that we need the 2 doses. And the other thing is we need to make sure and monitor individuals to check for immunity and check how the dosing regime's going. Certainly from the preliminary data from Israel, it looks very promising, and there's certainly a much reduced severe COVID-19 disease in the older adults who'd received 1 dose of vaccine. So the data looks promising so far, and we understand that we had to increase the dosing regime to ensure that the truly very, very vulnerable individuals were vaccinated and protected. Brian, do you have anything to add to that?
Yeah, just one thing really, which is that from the immune response point of view, increasing the gap between the 2 doses doesn't mean the second dose won't work properly. In fact, if anything, it might be better in terms of an immune response, in terms of making antibodies, to leave a longer gap. There's some data already from the Oxford vaccine suggesting that a longer gap is actually even a bit better in terms of the second booster dose providing higher levels of neutralizing antibodies. So that from that point of view, there's really not an issue at all. It's really, but I think it's worth saying that you should wait until you've had the second dose to feel like you have fully been protected against disease. But from in terms of how well the vaccines actually work to boost the immune response, it doesn't seem to be a problem to increase that gap.
Great, thank you both. So, you know, again, reassuring, and as you say, Emma, we absolutely need to be measuring what's happening in the community and in the real world so that we can optimize those the vaccine program going forward. So great questions, everybody, for submitting your questions. Now we've got a couple on questions on specific areas of health. So the first one is around immunosuppressed patients. I mean, firstly, should they be getting the vaccine? And secondly, well, how effective will that vaccine be in immunosuppressed patients? And the second one is lots of questions around, well, is there an effect on fertility following vaccination? Brian, do you want to go first and maybe tackle one of those, and Emma can tackle the other?
Yeah, for sure. So in terms of immunosuppressed people, definitely they should be getting the vaccine. That's absolutely clear, and that's the advice from the MHRA, is that those people should definitely be vaccinated. In terms of how well the vaccine works in those individuals, that's quite unknown and quite case by case, because there's lots of different types of immune suppression and there's lots of different context of that inside that question. So we don't know in all of those cases how well it will work, and there's lots of ongoing studies to try to find out, to try to monitor those people. So they should be very careful, but they should definitely get a vaccine for sure. In terms of the fertility question, Emma might be able to answer in more detail, but basically there's no effect on fertility whatsoever.
That's exactly right. Yeah, there's no link to the vaccine causing infertility. There's no reason for someone who is breastfeeding or who is pregnant and is recommended to have the vaccine not to have it. And actually, it's looking like the data suggests that those individuals who are pregnant and get COVID-19 get more severe disease than those who are not pregnant. So I think it's becoming increasingly evident that the vaccine should be recommended for anyone and everyone.
Brilliant, thank you. I'm conscious of time, but I'm afraid this is far too good to finish at 12:45. And as we've got you both here, I'm hoping you can stay on for another 5 minutes or so. Would that be okay?
Sure.
No problem at all.
Excellent, thank you. Thank you, and hopefully people can stay on to the end of the webinar as well, which would be wonderful. So we've had some questions in about developing symptoms to the vaccination itself. So, you know, questions like, why do people develop symptoms? Why do they develop different symptoms following vaccination? And if you don't get any side effects, does that mean the vaccine hasn't worked? So Emma, do you want to take this one first?
Yeah, sure. So the most common symptom of the current vaccines, I think, are fever, tiredness, and sore arm. These are all signs that your body is recognizing the injection, the vaccine, and is responding by generating an immune response. So if you get a redness and swelling at the site, that's just a sign of inflammation, sign that the body is recognizing something foreign's injected. So, you know, whenever people ask me this, I always go, well, it's a good sign. It shows your immune system's recognizing it. But conversely, if you're lucky enough not to get any symptoms, it doesn't mean that the vaccine isn't working, and just perhaps, you know, you're harder and not getting any of the side effects. Do you have anything to add, Brian?
I mean, actually, Brian, can I ask you a supplementary question? Because there was one that came through. It's like, well, will side effects differ for the different types of vaccine as well?
So in general, probably a little bit, but not very much. There are slightly different side effects, but they're a generalized, like Emma was saying, it's a generalized sort of inflammatory response that some people get to a greater or lesser extent, and those symptoms will be very similar between vaccines. And that's the same for other vaccines as well. Like the flu vaccine, some people respond and feel a bit ill sometimes, even after the flu vaccine. That's the body's response to that vaccine. That can happen to some people. It doesn't mean it is working better or worse. There's no correlation there. There's no relationship between how bad you feel or how well you feel and how well the vaccine is working. It's just a slightly different response.
Brilliant, thank you. Nice to have that little musical interlude as well.
Sorry, it's my doorbell, yeah.
Brilliant. So, I mean, there are now a couple of questions just to sort of end the webinar on, which is kind of more about how to have the conversation with patients, because, you know, that can sometimes be quite challenging. And so we've had some questions coming in about, well, how can we acknowledge some of the unknowns about these vaccines? Because, you know, they haven't been around that long, but how can we acknowledge those unknowns without scaring somebody or scaring the public, like, for example, long-term effects of these vaccines, which of course we don't know beyond the time that they've been trialed. Emma, I mean, have you got any tips on how people can have those conversations?
Yeah, I mean, again, I just got to be honest and say that the studies that I've carried out we've probably had a year's worth of data now, and it looks very promising, and there seems to be no long-term effects for sure. But we also wouldn't expect there to be any long-term effects of vaccines. All the other previous vaccines that we've designed effectively haven't had any detrimental long-term effects. So I think, you know, we wouldn't necessarily expect it, and our other vaccines that we've come up with haven't had it. But you've always got to accept the unknown, and really, I think, you know, a vaccine is more preferable to severe COVID-19 disease and hospitalization and worse.
Thank you, Emma. And I mean, Brian, Emma mentioned about other vaccinations. I mean, are there any examples that you can give for our health worker audience to be speaking to their patients about other vaccines that have been around for years, if not decades, that are based on similar technologies to the ones that have been developed for COVID?
So that's a good question. So in this case, there aren't really, and I think most of this conversation about long-term effects is around the mRNA vaccines, which is extremely understandable because they are in the clinic, they're new, and so that in itself is an unknown about what might happen in the long term. So Emma is correct. You have to acknowledge the fact that a new medicine has as some potential for long-term issues. But on the other hand, there's a really powerful conversation to be held about the risk versus the benefit, where the risk of there being a long-term problem is extremely small, and the benefit of taking the vaccination is great compared to the problems of COVID for yourself, but also for the whole society, the whole community, whereby we really want to eliminate SARS from our communities as much as possible. And the way to do that is to get as many vaccinations as possible. And so not only are you helping yourself, you're also helping other people in the community by getting vaccinated. So those kinds of conversations absolutely have to acknowledge the concerns, absolutely have to acknowledge the fact that there are some unknowns, but the unknowns come with very, very tiny risks compared to the benefits of what we're doing.
Brilliant, thank you. And so we will let you go very soon because you have got other things to be getting onto, incredibly busy people. But final question that I'll pose to each of you, so please do respond in your own way to it. But we have an audience of hundreds of healthcare workers watching today. How important is the role of a healthcare worker in answering the questions that the public and patients have on vaccinations and addressing some of the concerns that people will be raising? As you said, Emma, at the beginning, a question doesn't mean someone is anti-vaccine. It's a genuine question. So how important is the role of the healthcare worker in having those conversations? Emma, do you want to go first?
I mean, healthcare workers are critical because they're the only people who can go out and speak to so many people and engage and discuss with them about how vaccines work and how immunology works and about why we want them. We as scientists don't have the ability to go out and speak to everyone. Whereas by disseminating the information to you guys, it means you can now go out and have those conversations and give people the information they need. Because right now it's currently very difficult to find out what is misinformation and what is the true facts. And we know that individuals believe everything, or pretty much everything that a healthcare worker tells them. They know that they're speaking from fact, and that is really, really important.
Great, thank you, Emma. And Brian?
Yes, absolutely. And the role of healthcare workers is, as Emma said, absolutely essential in this process. Not only delivering the vaccines, but making people feel comfortable answering the questions and reassuring people. This is such a critical role. We know from the MMR scare 20 odd years ago that the impact of misinformation is massive, and the best way to combat misinformation is through lots of people talking to their friends, their family, their neighbours, in the clinics, to their doctors, to their healthcare workers, on a one-to-one basis, to talk to people, to reassure them if they have questions, and to engage with them, to have questions, to get those questions answered by people that they trust and by people that they feel will give them a good quality information. We're quite lucky in the UK, which is that people still have a good amount of trust in healthcare workers, in their doctors and nurses, the people who work in that environment, and their scientists as well. There's polls that suggest that the trust in healthcare workers and scientists is high in the UK. So there's a really powerful way to combat misinformation, and that's through having those direct interaction with patients, with the people in the community, and it's very important and very powerful.
Brilliant. Well, on that note, I couldn't agree more, but on that note, thank you very much, Emma and Brian, for giving up your time for the webinar this afternoon. Fantastic and fascinating insights into the science and what's been going on with the vaccines, and lots of your insights into the questions that have been asked. We've had a lot of thank yous and a lot of positive comments from people on the webinar. So huge thank you to you, Emma, and to you, Brian. And also thank you to all of you who have joined the webinar this afternoon. As I said, we've had hundreds of you on here and still on here. So it's been great to have you join us, and thank you very much for your questions. I'm very sorry if I wasn't able to ask the question that you posed, but I can assure you we've collected all the questions that have come in, And as we do at the British Society for Immunology, we will continually evolve and improve the resources that we've got on our website based on the feedback that we're getting. So we will take all of your questions into account, and if there's a way that we can answer them in the guide or on social media, somewhere on our website, then we will absolutely do that. So thank you for your engagement this afternoon. I'm also going to just a final plug again for our guide. And so please do have a look on our website at the guide. It would be great to get any feedback that you've got, but please do feel free to use that as you see fit. Hopefully it's useful for you. And we've also recorded this webinar. We did get Emma and Brian's permission beforehand, of course. So that is recorded and it will be put on our website in the coming days. So please do feel free to click on and have another view and also promote to your colleagues as well if you found it useful. It will be there, free to access. Please do make the most of that. So that just leaves me to say a big thanks again to our speakers. Thank you, Emma. Thank you, Brian. Thank you to all of you for joining us, and genuinely, without wanting to sound too cheesy, thank you for everything that you are doing as our healthcare workforce. You have been and you continue to be amazing during this pandemic situation. If there's a tiny little bit that we can do at the British Society for Immunology to help you in your roles, then we will try our very best. So hopefully the webinar has been useful for you this afternoon, but it'd be great to get your feedback on that as well. So thank you, everybody. Well, I'm sure we will see each other again in this virtual world at some point. Thank you, Emma. Thank you, Brian. Thanks, everyone. Speak to you soon.